Assalamualaikum warahmatullahi wabarakatuh. Hi, I am Dr. Pratiwi here. Welcome to my class. Today we will talk about growth and development of the tooth. Please enjoy. In this diagram of the mandibular arch, illustrate that two sets of teeth develop in the human. A primary or deciduous dentition and a secondary or permanent dentition. As we can see here, permanent teeth drawn in red are successional teeth. They arise lingual to the primary teeth and replace them. The permanent teeth, outlined in black with yellow centers, are the permanent molars that develop posterior to the primary teeth. They do not have deciduous predecessor, or they succeeded by successional teeth. Three types of teeth constitute the primary dentition. There are incisors, canine, and molars. Three types of teeth constitute the secondary dentition are incisors, canines, premolars, and molars. The first teeth to develop and erupt into the oral cavity are the deciduous mandibular central incisors at about 6 months after birth. The last primary teeth to develop and erupt are the second molars at about 2 years age. The first permanent teeth to erupt are the first molars at about 6 years age. The first, second, and third permanent molars erupt at about 6 years intervals, at 6, 12, 18 until 25 years of age respectively. In this photomicroscope, we can see here that dental lamina is a band of epithelial tissue seen in histologic section of a developing tooth. The dental lamina is the first evidence of tooth development and begins in humans at the 6th week in utero or 3 weeks after the rupture of buccopharyngeal membrane. At the site of each future tooth, the epithelial cells of dental lamina at A proliferate to produce swellings. These swellings along the shelf-like dental lamina are referred as primary dental organs or enamel organs or tooth buds at B. This is referred as bud stage of morphogenesis. There is a condensation of underlying mesenchymal cells at C in the region under the central aspect of the bud. This region of the tooth bud will continue to invaginate, giving a tooth germ a cap-like morphology. Please give attention the beginning of the formation of the alveolar bone socket at D. Next, we will learn about the capstage. As we can see here, the dental organ in the capstage consists of inner dental epithelium at B, which form the inner lining of the cap. Outer dental epithelium at C forms the outer lining of the cap, and the stellate reticulum at D occupying the area between the two epithelial layers. The outer dental epithelium and inner dental epithelium are continuous at the cervical loop at E as the rim of the cap. The condensed mesenchymal cells at F capped by the dental organ will form the dental papilla. Another condensation of mesenchyme that surround the dental organ and merge with the dental papilla is the dental follicle or dental sac at the G in this photomicroscope. The dental organ and the dental papilla and follicle together are called the tooth germ. Since at this stage all the formative tissue are present, will give rise to a complete tooth. Now we are in bell stage of morphogenesis. This tooth germ is further developed is seen in this preceding image. The cells in the cervical loop area at A proliferate and causing the rim of the cap growing deeper into the connective tissue. This transforms the shape of the dental organ from a capstage to a bell stage. The tooth germ in this section is considered to be in the early bell stage. Stratum intermedium at E, intermediate in position between the stellate reticulum and the inner dental epithelium cells. On next chapter, we will learn about tooth development. This image is from the region of a developing tooth where odontoblasts at A are differentiating from the mesenchymal cells at B of the pulp. Identify the following in this image: predentin, inner dental epithelium, stratum intermedium, and stellate reticulum. The differentiation of odontoblasts is a result from the influence of the inner dental epithelium cells. Secretion of predentin. As the odontoblasts continue to differentiate on the periphery of the dental papilla at B, they begin to secrete a product between themselves and overlying cells of the inner dental epithelium. This product is predentin. Predentin is converted to dentin by mineralization. The central region of a bell-stitched tooth germ is in the center of the field. You should be able to identify inner dental epithelium, outer dental epithelium, stellate reticulum, and stratum intermedium. On dentin secretion, identify the following structure in this high-power image of the peripheral region of the pulp. Odontoblast at B, the light pink is predentin, the darker pink is dentin, and enamel at E. Note that the odontoblasts at B vary in height and appear to be pseudostratified. Columnar-shaped odontoblasts are actively secreting predentin as an organic matrix. The deposition mineral in this organic matrix convert predentin to dentin. The odontoblasts move away from enamel as the predentin is deposits, but they leave a cytoplasmic or odontoblastic process with a tunnel or mineralizing dentin called dentinal tubules. This is a higher magnification of the region of the dental organ near the cups from the preceding image. The cells of this region are actively involved in formative activities. Identify the following: stellate reticulum, stratum intermedium, inner dental epithelium that are differentiating into ameloblast. To the left of the image, dentin, differentiating odontoblasts from the cell at the periphery of the dental papilla and the dental papilla. This tooth is further advanced in its development. The columnar inner dental epithelium cells producing enamel called ameloblast. The enamel is the deep purple staining layer forming a cap over the reddish dentin layer. Enamel is deposited between the ameloblast and the dentin. Enamel formation does not take place until some dentin has been formed. Note that an outer dental epithelium, stellate reticulum, and the stratum intermedium can no longer be distinguished in the region over the ameloblast. The condensation of these layers bring capillary beds close to the ameloblast, which no longer can count receiving nutrition from the capillaries in the dental papilla because the intervening layers of dentin and enamel. Next, the image close to the cups identify the purple staining enamel overlying the broader layer of the pink staining dentin. Numerous capillaries lie adjacent to the outer surface of the outer dental epithelium. The dentino-enamel junction is indicated by the asterisk. This is a cross-section through the crown of a tooth. Lines of ratios appear like growth rings in a tree stump. Enamel is harder toward the free surface than toward the dentino-enamel junction. Enamel hardness depends on the degree of mineralization, the orientation of enamel rod, the orientation of the crystallite within the rod, and the distribution of metallic ions which occur in trace amount. Hardness of the enamel surface may be increased by the presence of fluoride ions. Imperfectly calcified matrix of dentin situated between the calcified globules near the dentinal periphery, also called interglobular space or interglobular dentin. Odontoblast processes may not withdraw quickly enough back across the dentino-enamel junction after touching the preameloblast and remain stuck in the enamel. These projections are referred to as an enamel spindle. This is a photomicrograph of an erupting tooth. The formation of the crown of the tooth has been completed, and root formation is starting at the region of the cervical lobe or epithelial diaphragm. In the human, root formation continues during active tooth eruption with deciduous teeth. It is completed in 1 to 1.5 years after tooth appear in the oral cavity. In permanent teeth, it takes about twice as long for root formation to be completed. At the cusp end of the tooth, the reduced enamel epithelium fuses with the oral epithelium during eruption. This is a ground cross-section of an enamel lamella. Enamel lamella are fissure-like defects that extend along the longitudinal axis of the tooth down to the dentin layer. True lamella may contain debris from the oral cavity. Enamel spinel is short, linear defect found at dentino-enamel junction and extend into the enamel, often being more prevalent at the cusp tips. Enamel tufts are hypomineralized ribbon-like structure that run longitudinally to the tooth axis. In this photomicrograph, please identify the capillary and nerve plexus on the dental bulb. Secondary or cellular cementum overlies the primary cementum layer. Cementocytes live in the lacuna of cementum, and cementocytes are nourished by diffusion.
Hi, Dr. Christian here. Today we are going to discuss about periodontal membrane. As we know, the periodontium is defined as those tissues supporting and infesting the tooth and consists of cementum, periodontal ligament, bone lining the alveolus, and that part of the gingiva facing the tooth. Proper functioning of the periodontium is achieved only through structural integrity and interaction between these various tissues. Let's see for the slide number 1. Let's make it large. This is alveolar bone. And then this is gingiva. And this one is the root lined by cementum. This is cellular cementum and acellular cementum. We go to slide number 2. The dental alveolar crest fibers and the horizontal fibers of the periodontal ligament. See, this is dental alveolar crest fibers. For A, and then this is horizontal fibers for B. This is alveolar bone for C. Okay, we go to slide number 3. The insertion of periodontal fibers into alveolar bone and cementum. The horizontal line in the bone and cementum represent the Sharpe fibers. See these horizontal lines that embedded into cementum or alveolar bone represent the Sharpe fibers. The fibers are more numerous but smaller at their attachment into cementum than at the alveolar bone. Okay, we go to next slide. The initial cementum formation. The first increment of cementum forms against the root dentin surface. Epithelial cell rest of molasses. Remnant of the root sheath. You can see the cell rest of molasses in the periodontal membrane. See, this is rest of molasses. We go to last slide. This slide will show us all about periodontal membrane part. See, in here we have 7 items for showing. Number 1 is Sarpe fibers. See, this number 1, Sarpe fibers. And then number 2, epithelial cell rest of molasses. This one. And number 3, we go to cellular cementum. This is cementum with the cell. And then we have the number 4, alveolar bone. See, this is all alveolar bone. And then number 5, periodontal membrane. This part is periodontal membrane. Mostly it consists for connective tissue. And then the number 6 is dentin. And the last, number 7, dentino-cemento junction. Okay, that's all for today. See you next time.